to the Bioinformatics Core second webinar, where we're going to be talking about normalization, artifact detection, and the outlier detection as well, and then eventually also correction. This is the second webinar in the series. The first one we covered a little bit about how to access data in Synapse, how to access the C4 data, so the cell characterization and the genomic data that comes from that, both metadata and genomic data, methylation data, microRNA data. Today we're going to take a look at one specific data set. We're going to look at the expression data. These are the newest expression data that just came out in the last week, so at the end of August 2014. And we'll look at specifically the stem cell state data for that. And we're going to look at the quality of the data and see if we find any artifacts in it or batch effects, etc. Before I go into that, I would like to go and give a quick introduction to something that I'm going to be leaning on very, very prominently. And the idea of that is the singular value decomposition, also known in special cases as the principal component analysis. The idea behind singular value decomposition is if you have a data set, you're trying to find what explains the data the best, i.e. what is the thing that is varying the most in the data. And so in this example right here, I have basically what an experiment could look like if you only have 2 samples and you only have a few, about 80 genes or something like that. So you imagine you do an mRNA expression. This could be an array-based, or it could be a sequencing-based, and you captured a gene expression for, say, 2 patients. More usually, you will have many more samples than that, and of course, thousands of genes. But then if you plot the value of each gene for each of the 2 patients in this case, you might end up with a scatter plot as the one that you're seeing on the right. And the reason I only chose 2 patients is because it makes it very easy to plot. But in reality, you're working when you have hundreds or dozens of samples, at which point the plotting it becomes very difficult because you have to have one axis for each of those. And if you look at this data by itself, you can see that it actually falls along a sort of a cloud of points that goes up and diagonally across the x and y axis. And the singular value decomposition, or the PCA does, which is normalized form of the singular value decomposition, is that it actually finds those directions of variation. Now, at the next slide right here, so mathematically, say that the matrix that we were looking at before, which had the expression values, is D. It turns out that you can always write any matrix as long as the product of 3 different matrices: a matrix U, a matrix sigma, and a matrix V transpose. And you can always reconstruct your data into these 3 submatrices. And algorithms for doing this that have been around since the 60s. It was initially invented in the 1880s, actually, but there were no ways of calculating it efficiently. But the interesting part about these is the matrices U, sigma, and V have very specific properties. One of those properties is that each of the columns in U are orthogonal with each other, which means that they're independent. And the values in sigma are 0 everywhere except the values on the diagonal, and those are the ones that are equivalent to, they're called the singular values, and they kind of represent how much each of these vectors in U capture in the data. So it's a measure of information contained in each of these vectors. And the third matrix, V transpose, is again an orthogonal matrix, which means that each of the rows in this matrix are independent of each other. So by applying this algorithm SVD, you basically can reconstruct what are the largest directions of variation subject to the condition that they are orthogonal to each other. And diagrammatically, I've shown this for the data set that I showed in the first slide. Basically, the first red arrow, the one that goes diagonally from the left to the right upwards, that captures most of the variation in the data because it follows the shape of that cloud of points. And then the second vector here, which would be the principal component 2, basically is the one that is orthogonal that then captures the remaining information in the data and remaining variation in the data. And so this is what we're going to be working with today, and specifically we're going to be looking at these vectors that are in the V transpose matrix. The reason it's interesting is if you have a batch effect, it would affect the majority of the data, and so it would correspond to a large portion of the variance. If it doesn't correspond to a large portion of the variance, i.e. it's not one of the more highest principal components or singular vectors, then it's not going to be a strong component of the data. One thing to note is that this is assuming that the effect is on a global basis. You could have a batch that is affecting only a subset of genes and a small subset of genes, at which point the singular value decomposition or the PCA would not be able to detect it. So unlike last time, I'm going to go over and I'm going to actually run code, and we're going to look at fetching data, doing the singular value decomposition, and try to identify what these patterns are. And the way I'm doing this is I'm running everything in Python, so it's a Python notebook, and I will leave this one available, and I'll attach a link to at the bottom in the comments to this video, so that you can find where it is, and you could download it and rerun it yourself and reproduce the results. It actually is a normal Python code. The way I'm presenting it is specifically in a Python notebook, which is an interesting and easy to use interface to Python, but you don't need anything special to run the code. To bring in my Python code, and it might be a little complicated to begin with, but most of this stuff in the first stage right here, the first part is basically me importing all the things that I'm going to need. So these are packages that are going to help me, like such as statistics packages for doing modeling, Synapse client so that we can fetch the data and upload the results. And then below that, I have defined some constants which describe where the data is located, and I'll show you again what that looks like. And then lastly, what I do here is I just log into Synapse so that I get access to this. So I control-click to run this, and it runs now. It's actually connecting to Synapse. It says, "Welcome, Larsson Omberg." I'm now logged in. And the next thing to do is I'm going to fetch the data. And this basically this command right here, I've given the ID that I gave above here for the expression data, and I just do synget expression data. What this does is it downloads it for me, and then the next step I'm doing right here is I'm reading it into memory, and I'm using a pandas data frame for this, which is similar to a R data frame. And then I am also fetching the metadata and reading that in as well. And then it turns out the metadata has some rows that are duplicated, so I get rid of those. And then I filter the data for those that fail QC, and this could be QC that has been determined prior based on the alignment, and lines, so these are cell lines that have been considered to be bad. And then I get rid of, and then I also only keep those that are stem cells. So there's some samples in here that are from tissues, but I'm not interested in those. And then there's a lot of information in the metadata that is not relevant for this analysis, so I drop those from the metadata to make the metadata easier to understand. And then last thing I do here is I basically find those columns in the metadata and the expression data that are the same, and I extract only the data that we have metadata for, and both that expression data and the metadata. So I run that, it's now downloading it from Synapse, and then doing the filtering, and eventually also the matching of pairing up the samples. Now, I'm going to add a line here just to make it a little bit easier, so you see what things look like. If I look at metadata, for example, like that, we can see that metadata is a table. It has information about the cell lines, what they are, a UID, which is identifying of the canonical cell line. So this is the cell line differentiated in mesoderm. We can see where it derived from, and this is information such as the origin and how it was processed. But what I want to do first is data set, the actual the data that we download, the expression data is very large. It's 57,000 different locations, expression for 57,000 locations. Most of this data doesn't have very much variation, is not interesting, and to make the computations easier, I'm going to throw out all of the probes, all of the genes basically that have variation across the standard data set, which means I log transform the data, and then I check for the standard deviation, and it has to be larger than 0.5. And then here I extract those. This is again just filtering down the data to smaller size. And if we can actually, at the end of here, we can actually look at expression data, print the shape, which is the size of it, and we can see that it was 57,250, and it actually changes to expression data, high variance, and reduced to 12,700 genes by 189 different ones.
Larsson, a really quick question here. You chose, since this is FPKMs, correct, as the expression values in this matrix?
That's correct, yes.
So by adding one, you're adding a single FPKM to each value prior to log transforming. That's correct?
That's correct.
Isn't that too much?
I don't think that's going to affect the results significantly. The reason I'm adding one is because I'm doing the log transform, and log of 0 is undefined, and so it's just to get it slightly off from. And when you do the log transform, that transforms to 0.
The only reason I asked is that if you had, and theoretically you're looking at genes with probably an FPKM greater than 3, for example, is that you will impact the fold change, but it may, with higher expression genes, it's going to be less of an issue with adding one. But I just wanted to bring that up for folks that were listening, as that value is potentially important to think about in the future.
That's a very good point, and perhaps the more careful solution would be actually to perform a second filtering on FPKM as well. So we're only looking at those genes where the FPKM is above a certain threshold for a certain number, and then we'd reduce the effectiveness of this. And again here, I'm now doing with the new high variance one, I'm doing a log transform, and then I'm going to normalize each sample. So I'm doing the log transform since they're FPKM, which are close related. It's always going to be a positive number, and so I'm log transforming them to get them, and I'm mean centering them basically to get the variance from, so it's around 0, and then I'm dividing also by the standard deviation to basically turn these into z-scores. So that each gene is now represented by a z-score. And this is just to make it so that the statistics underlying the singular value decomposition becomes more similar to a PCA. So I did that actually by column. So this is standardizing each of the experiments, and then I'm going to do the same thing by standardizing across each of the genes as well. And then after that, I'm going to extract only those genes that are stem cell. So I'm going to ignore all of the differentiated states. Again, we can repeat this analysis without doing this filtering first, and we'll basically be looking at all the different states, so mesoderm, endoderm, ectoderm, embryoid body, and stem cell. But we're going to look at the stem cell so we can see more of the variation that's associated with one state, because most variation otherwise will capture the differences between the different states. And lastly, I'm going to perform a singular value decomposition. So I pass in my data matrix, and I get back these three matrices, U, sigma, and VT. And then last thing I'm going to do is I'm going to plot the first singular vector. So these first singular vector versus the second singular vector in the first plot, and then a second plot with the first one versus the second one, so we can see what these look like and see what the variation is. And I'm color coding by anything like that. But what you see, what happens here is that the majority of the variation in the data, so PC1, actually responds to the difference between one sample in the set and the bulk of the samples over here. And actually, if you look, it captures both in PC1 and PC2. This one sample is a far outlier based on this, and actually is the majority of the variation in the data. And so we could go in and try to figure out what this means by looking at which genes are associated with this signature. But I'm going to say that this sample should probably fail the QC analysis as well. And I just recently looked into this, and it turns out that this one has a very strange karyotype as well. And since we look at the PC1 right here, this sample is the only one that has a negative value. Everything right over here is positive. So I can find which one I'm looking for by just looking at the first singular vector, which values are less than 0. This would be the sample that I'm interested in. I'm going to look, and I'm specifically going to look inside the metadata. That's called M right here. And so you can see this is the sample that we're looking from. So it's SC12040. It's a fibroblast, and the tissue of origin says foreskin. But then if you look here at gender, it says female. So there's probably something wrong with the classification of the sample to begin with. So we move forward, we're actually going to get this sample. So I'm just going to say, take the data that I have, drop the sample, store that, drop the sample from the metadata as well, and then to perform a new singular value decomposition. So this basically where it detected an outlier. And here I have a convenience function that I have written, and I can share that as well, which basically does the same step as above. It does the singular value decomposition, but it also looks at the covariates and finds for each singular vector which ones are the most correlated with one of the singular vectors. So if I run this, it takes longer because it does a little bit more work on it. And it creates a bunch of different figures. So actually the first figure right here is this V transpose matrix. So this is all the singular vectors that we would be looking at. And then on the second field here, you actually show what the variance or the amount of information captured by each of the singular vectors. So this is what's in the S matrix. And then this plots the first singular vectors on typically the first few rows over here are plotted out instead of just as a heat map. Then below we see the same kind of information that we saw before, so the PC plots where we're plotting PC2 versus PC1. And here in percentages, you can actually find out what the kind of amount of variance captured by each one. And what we see here is that the first one captures 46%, and it's associated with high confidence donor ID. So it turns out in this data set, if we were to look at the metadata, there are multiple samples taken from the same donor, sometimes different turned into stem cells using different protocols, but they're the same individual. And so that has a strong effect and explains most of the variation. And then second PC here is associated with UD, which is basically the cell of origin. And the reason is, sometimes we have multiple repeats of the same cell. So that's a good sign that we're seeing that as a strong PC here. So in all honesty, if we then go down further, we can see that PC3 again is associated with these cell lines, so it's, again, and very strongly so, with a p-value of 10 to the minus 19. And this is done using a able test, basically a chi-square generalization of it. And then you can hear all the way at PC4. Here we're seeing an effect of lab. So this is very good news. So basically we're seeing that donor is the strongest association in the data, and then we have a little bit of information lines. But then lastly, lab of origin has an effect as well. Now, let's assume that we don't want this donor effect to actually be in the data when we do downstream analysis. We can include that in a model when we're trying to figure out differentially expressed genes, etc. But we can also correct for it up front. And what I'm going to show here is two different ways of doing that. So one of them is we've done a singular value decomposition where we said the data set is equal to this product of these three matrices, where the first vector is highly correlated with donor effect. So one way I can do is I can basically say, well, remove that singular vector from the data set and recompute the data matrix with that vector set to 0. And we'll basically say whatever was captured by that first vector is now no longer part of the data. And so the way I do that is I have that my matrix S. So I set the first singular vector because it was the first vector that contained this information that was associated with donor. I set that equal to 0, and then multiply my U matrix with my S matrix and my V transpose matrix to reconstruct a data set where the first matrix in U and VT are now not a part of the data set because S0 is equal to 0. And I'm going to do a new SVD, which is basically the SVD without that information in it. Wait for that to execute, and we'll get the same figure. What you'll notice, though, is that there's been a drastic change in the distribution of singular vectors. And if we look at the PC plots, we now see that PC1 is associated with UID, which is the cell line, which is good because we have repeated runs here. Second one is also associated with this. And third one is now originating lab. So more or less what happened is that the second PC moved into the first place, third PC moved into the second place, etc. But what we see here is now we have a data set that is stripped of this. Alternative, so if we step back now and we say, is there other ways we can correct for this? And there's multiple ways, but one of the ways we can correct for this is we can also build a model and we say, okay, well, it's really donor ID that we want to correct for. The PC could be donor ID and also have other effects in it, so I don't want to correct for that one. I'm only going to correct for a donor ID. What you do that is you can do per gene, you build a linear model where you say the expression of the gene is equal to some constant factor beta 0 plus a variable B1 times whatever the value for the donor ID is, and then an error term. And it'll basically tell you that you're trying to explain the gene expression based on this donor ID, and the error term is whatever is left over after that after you included the donor ID. And if we look at this error term, or the residual of this model, that will basically be the effect that is left over after you've corrected for donor ID. And what we do here is we keep our, I'm going to create a new matrix, which are these residuals, and then I'm going to go through these, the range of all of the genes, and for each one of those, I'm going to build a model. So here I'm just building the data set that I need, and here I'm building a model. So I'm saying that the gene expression is proportional to donor ID. Then I fit the model, and I extract the residual of that model. And then I store the residual of that model in this new data set that I have dnorm. And then at the end, I'm just going to put these all together into one large matrix and throw away all the samples where there's no information. And since not every sample has a donor ID, some of these samples will not disappear from this. And then I'm also going to remove, since I had thrown away some samples, I'm also going to throw away those from the metadata, so I have a coherent data set between the normalized and the. And if I execute that, it's now going to go through every single gene and do these models. It actually takes a while to do because it has to do a single model 12,700 times. And before, as it's running, I'll mention that the way this will not give you exactly the same result as when we did the normalization using the setting one singular vectors to 0. And the reason for that is that the we're now specifically only looking at donor ID. I said that already, but I'll highlight that. And we're not looking at the singular vector, which could be an effect that captures donor ID and other effects as well. And this is run. We're going to perform a singular value decomposition to see what this looks like. And this is still running, so it'll be a while.
While that's running, I was just quickly curious. So you commented out also. So you're going to visualize. It looks like a box plot by gender to the gender effects after you remove the donor effects. Is that correct?
You know what? It should have. I didn't mean to by gender. I meant to by donor ID.
Okay.
And the reason, yeah. And I pointed it out because this would generate a new box plot for every gene. So I was doing this to see what the model looks like for each one of these, and you know, I was running it just a few at a time. So running the first 10 just to see what the model looked like. It wouldn't work if I did this for every single gene.
Okay.
Make sense?
It does.
I apologize. I thought this would actually run a little bit quicker. In the meanwhile, I can actually go over to Synapse and show you what the data set looks like and what we're extracting. So we'll pull up an instance of Synapse over here. I meant to do this earlier, actually, when we're fetching the expression data and what it looks like when we're fetching that. So we'll go to Progenitor Cell Biology Consortium, and we'll go into the files. Actually, we can go here into Download Data. And the file that we're actually looking for is this summarized expression call right here. So what we downloaded is this data set right here. And you can see there are multiple versions of this file. And if this is version 2, we can actually end up going like that, and we can see that there's even more versions than that. And here also there's information about how this data set was created. Let's see how it would happen over here. So yeah, finished running the model, and then we've done a new SVD on this. And again, what we see here is in the previous one we corrected using a singular vector. The first one is now associated with a cell line, second one with a cell line, third with a cell line, and fourth with a cell line. So it's worth noting that the p-values here are really, really low. One thing to look out for when you're doing normalizations like this is if you do too much normalization, you can actually whitewash the data where you actually take out all the signal, because there could be correlations, for example, between donor ID and the signal that you're actually looking for, such as reprogramming and stuff. So I'm not saying that this is the right kind of normalization to do on this data. Last thing we're going to do is we're going to store this in Synapse. So the idea is basically we pull data in, and if you do an analysis on it, you can pull it up, you can store it back to Synapse and share it, and then indicate what you've done with the data. So the first thing we're going to do is we're going to store this code that we've been running. So store send something, and we're going to store a file, and I'm going to store this file that I've been working on right here. So this file is called bioinfo webinar demo 2 batch detection pi. You can see that up here. And I'm stored inside a folder. This is the ID of the folder. And then also going to save I'm going to save the normalized form of the log transform stem cell data. This is the one that we just generated. And I'm going to store that again on Synapse. I'm going to also store the normalized data set. So this is the data set that has been filtered down to the same set of samples. And then last, I do two things where I store them on Synapse. And then just to show, I'm going to open them up on the web as well. So this now writes the files to disk and pushes them up to Synapse. The progress bar that you see there at the bottom is the progress of storing them on Synapse. And so that's actually storing data sets, so it's a little bit larger, takes a little longer. And then the last thing is going to be to store the metadata.
Larsson, quick question. It seemed like gender was not an issue from the PC plots that you showed. Why do you think that is? Typically, because we've seen gender as a major driver in the pluripotent samples.
Yeah, I'm fully aware of that. So I know we've seen that in previous looking at the data. I'm wondering if it's because of the larger sample set that we're looking at now. And I think this is a conversation we should probably have offline, just the two of us. I'm not seeing a gender effect as before, and I'm also not seeing an effect that we used to see where it was related to pseudogenes and non-coding expression versus gene expression. So this data set looks very different than it has before the addition of the last 50 or so samples to it. So back in Synapse now, this is now the data set that I had generated using this code. So this is something you can share with one of your collaborators. We have information about how it was generated, so I stored the code. So if I click on here, I can actually see the code that was used to generate this. So there's code that we ran. If I go back, we can see that I used the metadata, the original metadata as input, and I used the summarized expression call as input. And it's also worth noting here, so I can do things such as add more descriptions if I want. I can basically go and add a wiki page to this if I want, so and add a description, and now I can actually add free text to describe what I did as well. So if you're using this to share with collaborators, it could be helpful for them. The last thing I'm going to do is to show that, which is something that has come up by request, is the fact that how do you work with Synapse outside of for example, Python or R, which is the fact that we do have interfaces so you can actually download things directly into Galaxy. So if you would prefer to do this kind of analysis in Galaxy, what we do is actually, I'm going to, before I do that, I'm just going to take this SynID of this normalized data, and I'm going to copy that, and I'm going to go over to a Galaxy instance. This is a Galaxy instance I'm running on my own machine, and you can install a Synapse interface through the tool shed. So I'm going to basically say, okay, well, I want to upload Synapse downloads. I'm going to fetch this data set that we just looked at. And so I'm going to pick the Synapse ID that I just copied over there. And you can put your username and password, but it's actually easier if you just log in by logging in using an open ID. You just click on this button right here, and it will open up a new interface that has logged into Synapse. And actually, you see, put in a session token here, which means that I'm now logged into Synapse. I'll put the ID in here, and I'll run execute the download for of Synapse into this. So you can see basically the process of this going. So the current job is currently running, and it's fetching this. We can, if we move back to Synapse, oh, there it is. It was already done. So here we see the data set that I had just uploaded. And basically now you can use whatever tools you would like to use in Galaxy to perform the same thing. So multiple interfaces, we could do the normalization in multiple ways, but I think this is one of them. If there are any questions, or I'd like to hand over if there are any questions, and otherwise, thank you very much for your patience. Thank you.
Larsson, I had a few questions. If
okay.
If somebody's continuing after you, I'm sorry, or this, or you're finished completely.
I'm finished. Do we want to stop recording, perhaps, or?
Yeah. It's okay to. I would like to a few questions that I wanted to ask related to what you just did.
Okay.
If it's okay.
Yes, please, please.
So the normalized the normalized expression values you just uploaded into Galaxy, those are effectively residuals. Those residuals are they're log2 values. Is that correct?
That's correct. Yes, or the residual of the log2 values. Yes.
And for those, if we were working with those residuals in an outside program, and we're now doing differential expression analysis, I don't know if we have time or if you want to go into that, if that's too much for today, to do a differential expression analysis with those values, for example, with cell of origin, for example. But you were doing an analysis, since you no longer have, the values are no longer similar, as I understand, to log2 FPKM values. Is there, interpreting the kind of relative magnitude of differences, how would you recommend going between those residuals and the original FPKMs to understand the kind of magnitude of expression and expression differences that you see?
Oh, I think I mean they should have the same distribution as the log2 of the FPKM. You wouldn't be able to say what the absolute expression differences are, but you can. As far as the distribution and the way that you would handle the statistics, it would be the same.
So in terms of calculating some kind of value, like a t-test p-value, for example, that should be somewhat assume that, for example, if the data didn't have any abnormal covariates that you had to control for, you would get similar p-values for the underlying data, whether you're looking at the residuals or at the raw data. But expression, the values themselves, the residuals, would be very different looking values than the log2 of the FPKM, correct?
That is correct. That's correct. Yeah. So if I were to do a differential expression analysis, I would probably use another linear model based on the assumption that it's normally distributed. So yes, that is completely correct. And if you don't want to move into the log2 FPKM standard, there are other normalization methods. So there's a popular tool called Surrogate Variable Analysis that is similar to what we did with the PCA, except it doesn't necessarily correct for the highest PC. It actually looks for portions of PC values. There's a new method that was just published in our archive, which looks at SVA seek, I think is what it's called, and I think that's based on looking at the raw counts or FPKM values. So you don't have to do the transformation because you're not assuming that the data is normally distributed, which is kind of what the assumption is when you're doing the standard singular value decomposition and the standard linear model that I was doing.
Something like SVA seek, you should be able to calculate full changes that are interpretable in a similar way to what you would get from analyzing, for example, cufflinks FPKMs. Is that correct?
I believe so, yeah. I mean, the point is, if the fold change specifically is interesting, another way of doing this is actually not to perform a two-step normalization. So, where you do a linear model that has the and then look at the residuals, you could also include basically a model where you have multiple variables included in the initial model, and where you do, like, instead of a t-test, you basically do like an ANOVA or something like that, where you basically have your effect that you're uncertain about, that you think is a batch, and then you have your true effect, and you basically fit those both at the same time.
So specifically working with, for folks out there who want to work with the C4 expression data, once they're comfortable with, for example, let's say they're wanting to compare their data set that they have to this data, it sounds like you'd recommend that they look at these covariates similar to what you've done, and potentially be aware of them, but not necessarily work within the residual space in analyzing the two data sets together. Is that correct?
I think if in the case where I had an external data set, I would probably do this kind of analysis across both data sets at the same time. So I would not do the correction first. I would actually combine the data sets, and look for the covariates that fit across the data sets, and then do it. Or I would do something even, if I could, which would be the absolute safest, which is to look at the data sets completely independently and identify, the signals in the data first, and then do comparisons on the signals. So then you wouldn't have to worry about that the distribution of the data sets are the same.
I think that's an important, challenging problem that a lot of folks analyzing this kind of data within the PCBC and outside have to deal with. So I think getting your opinion on that is very helpful to folks.
Mm-hmm. So I think my previous answer would be the opinion. So I would either do the data sets and then look at what the variation looks across. And some obvious things is maybe doing some very simple normalization first, not thinking about batches or anything like that, such as quantile normalization across the data sets or something like that, and then looking for signals that correspond to batch that might not be completely concordant with the 2 or 3 data sets that you're comparing. But absolutely the safest thing would be to look internally first, and then do across the data sets analysis of the signals instead.
And another question relating to Galaxy, these new features in terms of Synapse Upload, Synapse Query, Synapse Download, are those options that are automatically available in the latest instances of Galaxy if the servers that you're using are most up to date? Is that correct?
Not actually. So you have to add a specific toolshed to get to them. So they're not in the mainline toolshed yet, and I can't remember exactly to get to it. But I could write up a little description on how to do that. Putting on the spot, I wouldn't be able to answer it. But you would have to install them first as a toolshed, part of it from the toolshed.
So effectively, if folks have questions, they should probably either put on the PCB Synapse help desk, or to the Google Groups link that should be there very soon, correct?
That's correct. That sounds perfect, actually, yes.
Those are questions.
Thank you, Nathan. That's great.